Good evening. This is a spectacular view. This is like old home week or like family reunion. It is so wonderful to see so many faces. Welcome to Biotech 201. I am really glad you're here. My name is Neil Lamb and I lead the education programs at HudsonAlpha. Many of you know that and are well aware of that because you've been to many of these education programs. And welcome to this edition of Biotechnology, Biotech 201. We're going to talk about the human genome. Now, I also want to welcome not only those of you that are here, seats in chairs at HudsonAlpha, but the 100+ people that are following us online as well. So you in the room filled up these seats in 45 minutes in registration. All 300 of them, 45 minutes. And then we've got another 100 folks that are watching it live on the Ustream channel, and we are really glad to welcome you to Biotech 201 as well. Let me also stop and thank our partners, the Jackson Center, for the fantastic space that we're in. Isn't it wonderful to be in this room? How many of you were in the very first two Biotech 101s when we were crammed into the auditorium? It was great, but this is such a difference, isn't it? It's fantastic. And also ETV, who is doing all of the video and connecting everything to the Ustream channel. And so Keith Ward is over here in the corner helping make sure that our online folks get everything just like you are. So Keith, thank you. We're really glad to have ETV as a partner with us. So those of you that are in the room, hang with me for just a second. If you're a part of the online viewing audience, this is the screen that you're going to see. Let me just orient you through this right now. There's a link to the handouts down here at the bottom. Each week, a couple days beforehand, we'll put the new handouts there. This section in the center is where the video appears, and you actually have a couple of options. If you click the little arrows next to the word share, you can have the screen pop out so it's your own visual. You can see theater size, which will make it a little bit wider, or you can go full screen. So do whichever one of those you want. And then we also will have a moderated chat. You can click on the chat button up here near the video, and you can chat. We've got HudsonAlpha Education team members that are ready to answer your questions. And some of the questions, they will actually text up to me, and I'll take up here from the stage. And we'll also take questions certainly from the audience as well. All right, so here's what lies ahead for the next 4 weeks. Tonight we're going to talk about what is our microbiome, what is the finding of something called the human microbiome, and then we're going to talk about how it develops and early influences on the microbiome. And then in weeks 2 and in weeks 3, we're going to talk about diseases that are associated with the microbiome. Next week, we're going to talk about some new data with autism, different forms of behavior, cardiovascular disease, cancer, and aging. And then the following week, the third week, we'll talk about microbiome connections to obesity, inflammatory disorders, and autoimmune disorders. So we'll spend 2 weeks talking about all the different places that we have at least started to build some linkages with the microbiome and health and disease. And then on the fourth week, we're going to talk about how you tame the microbiome. So we're going to talk about the botics, antibiotics, prebiotics, probiotics, if you should be taking them, what you should be taking, if we even know if you should be taking them, do they make any difference? And this whole idea of can you intentionally adjust your microbiome? So that'll be a really interesting week. Everybody has the frequently asked questions. The American Microbiological Society put this together and put it out on their website about 3 weeks ago, which I thought was very nice of them to do that, knowing that we were about to have Biotech 201. It is chock-a-block full of great content and great information for you. So while I won't directly refer to it, there are things each week in our handouts that have linkages back to the microbiome FAQ. So I would encourage you to read it at some point over the next 4 weeks. I think you'll find that it's very well written, and it has some great visuals and some fantastic graphics as well. And for those of you that are online, the FAQ is under the link to download for the handouts as a PDF. Okay. There are some really important points that I want to make sure before we dive into any data, before we start talking about anything with the microbiome, I want to set the stage for you, and I want you to remember these things throughout the next 4 weeks. I'm going to do this visually. You do not have this in your handouts because these are all just pictures, and I wanted you to actually be looking at me in the screen instead of looking at your piece of paper. So, We are early in microbiome research, which is really exciting, but 90% of the papers about the microbiome have been published in the last 4 or 5 years. So you can think of this as the opening strains of the overture, if you are a symphony fan, or you can think of this as the first half of the first inning of the game. We are really early, which means a lot of what I'm going to tell you is one study, or maybe looked at 200 people. So recognize I'm giving you brand new stuff, but in no way is this the definitive, yes, we know that this is the answer, here's the connection. Which ties to my second point, which I think is the most important thing you might hear me say in introduction to the entire 4 weeks. Correlation does not equal causation. Say it back with me. Correlation does not equal causation. Thank you. What that means is just because you see two things that seem to be associated or going in the same trend does not mean that one causes the other. So here is a fantastic example. Here are two things. One of them that I'm measuring in bars, which corresponds to the percentages on the right-hand side, and one of them is this blue dot line that corresponds to the numbers on the left-hand side for the x-axis. So clearly you're seeing two different things that are declining as we move across years from 2006 to 2011. So this is a correlation. And if I told you that the blue dot line is the number of murders in the United States, you would be really interested to know what these green bars are, because maybe there's a connection. Well, in this instance, the green bars are the market share of Internet Explorer as a person's browser. Now, clearly, not using Internet Explorer does not make you less likely to commit murder. So, at least I don't think so. So, this is a correlation. This is not a causation. I'm going to show you lots and lots and lots of correlations. That does not mean they're causative. They may be, but I want to make sure you realize that what I'm showing you is a small number of samples, and we don't yet know for certain. So I need you to agree with me that you are not going to run out to your physician or to your children or to your pharmacist and immediately ask for the things that I'm going to show you. Can you agree with me on that? Good, because that leads to the next piece, which is do not try some of these. Do not try any of these at home yet. At some point, when we cycle back to this topic again, I might give you totally different advice. But right now, don't. Here's the other important piece, and I'm serious when I say this. We make decisions based on the data that we have at the time. Later, we may discover that those decisions had consequences that we did not intend. If I have conversations with you about early life exposures, about food preferences, about things that we do early in life, and correlations with disease, I am not pointing the finger of guilt at anyone. So do not point it at yourself. This is not meant to make you feel bad because you did or didn't do something for yourself or for your children. All right? This is information. There is no guilt and there is no shame allowed in Biotech 201. Now, your microbiome is the collection of bacteria, viruses, and fungi that live in and on your body. And in order for us to study that, we have to get that out of your body. We are going to have some conversations about some things that definitely would not be appropriate table talk. I am going to talk about parts of the body. I'm going to talk about biological functions. I'm going to talk about excrement and feces. I'm going to use appropriate clinical terms. I don't think it does us any good if I use cutesy language to talk about parts of our body. I'm not going to use outlandish, shocking language. I'm not going to use crude language. But I'm going to talk about parts of our body and the female reproductive tract. And I'm going to talk about feces from our gut and our intestine. Just recognize that that is what the microbiome is. And it's not intended to intentionally shock you or be crude, but I want you to be informed. So just recognize the language that we're going to use, and that we're going to spend a lot of time talking about poop. All right, last key point. We have spent most of the 20th century and part of the 21st century looking at bacteria as things that are the enemy that must be destroyed. That all bacteria are bad and that they cause death and pestilence, which is what this image behind me is titled, Death. I want you to try to strike the language of warfare out of your vocabulary for the next 4 weeks. The vast majority of our microbiome is your friend and is very, very helpful. So for example, these are some of the benefits of our microbiome. They help in digestion. Many of the bacteria that live in our body produce vitamins and nutrients that we won't get any other way. They help our cells grow and mature. We're going to spend a lot of time talking about how they educate our immune system. And when you have healthy bacteria in your system, they crowd out space for the dangerous bacteria. Yes, there are disease-causing pathogens. Yes, we do not want them. But I want us not to think about every bacteria as automatically bad. So those are the key points. Everybody ready? All right, let's go. So what is your microbiome? So your microbiome is the collection of bacteria, viruses, and fungi that live in and on a particular environment. Now, we're going to spend most of our time tonight talking about bacteria. In fact, for the next 4 weeks, we're going to talk a lot about bacteria. We just haven't yet been able to dig into fungi and viruses quite the same way. I hope to be able to add some of those pieces where we go, but the vast majority of tonight's conversation is going to be about bacteria. Every place you can imagine has a microbiome, has a microbial community. Whether we're talking about your gut or your skin or the soil outside your house or the water inside your pipes, there is a collection of bacteria, viruses, and fungi that live in that space. Make peace with that. It's okay. In fact, it's actually beneficial. We're going to spend a lot of our time on the human microbiome, but where we can, I'm going to pull in microbiome of other organisms or of other cultures and ecosystems as well. Now, how do we identify our microbiome? Historically, we did that by culturing them. So we would grow them. We would take a swab, maybe of the soil, or maybe of the water faucet at school, or maybe inside our mouth, and we would spread it on a little plastic dish that had a nutrient auger, almost like Jell-O on it. Who did these? This is like a high school or college lab. You plated out bacteria, and then you looked to see if it grew in red colonies or yellow, or and if you looked under the microscope, was it rod-shaped, or was it little tiny circular spheres? That was historically the way that we knew what bacteria were present. And based on that information, we figured out there were about 5,000 species of bacteria that we knew of, that we could classify. The problem with this is that a very, very small percentage of bacteria can be grown in those kinds of culture conditions on plates. So we knew we were missing enormous amounts of bacteria that we couldn't culture. So you have heard me talk a lot over the sessions, the Biotech 101 and 201 sessions, about next-generation sequencing. These are the machines at places like HudsonAlpha and other places around the world that very rapidly and very inexpensively sequence large swaths of DNA. People realized that we could use these next-generation sequencing machines to sequence bacterial populations. So I could take a sample of soil, I could remove the soil and keep the bacteria, I can break the bacteria open and extract their DNA, and then I can sequence the DNA of those bacteria. So from this point forward, almost every experiment that I'm going to tell you about, almost every finding that I'm going to tell you about, uses this technology and does one of two things. One thing, and this is still the predominant, the more common thing, is that they look at a specific gene. Remember, a gene is a recipe, a set of instructions. They look at a gene called the 16S ribosomal RNA gene. I think that's probably the last time you're going to hear me say that. Well, no, because I'm about to say it again. And so the sequence of this 16S ribosomal RNA gene is unique for two things. One, it's not present in humans. It's only present in bacteria, so it's a way to know I'm only looking at a bacterial population, not human cell samples. Two, this 16S ribosomal RNA gene, that was the third time, has lots and lots of variation in it. So in one bacterial population, it may have one certain sequence, and in another bacterial population, it may have a slightly different sequence. This is really beneficial because you can sequence through this gene, and based on what changes you find, you automatically know what bacteria are present. So it's like a bacterial genetic fingerprint. So most of the studies that we're going to talk about use some form of that kind of approach to look at a population, whether it's our gut or whether it's our soil sample or whether it's swabbing across your kitchen countertop and identifying what bacterial community lives there. The other thing that some groups are now doing is they're sequencing the entire genome, not just the 16S ribosomal RNA gene, and that's the last time I'm going to say that. They sequence through the entire genome of every single one of those organisms. That is an enormous amount of data, and as you might imagine, requires huge computational power to be able to separate out, well, this is the genome of a Staphylococcus, and this is the genome of a Streptococcus, and this is the genome of an E. coli. But the power exists to do those kinds of approaches. So this high-throughput sequencing approach now lets us suggest or estimate that there's over 1 million different species of bacteria in the world. Pretty big jump from 5,000 to 1 million. And it truly is a game changer. When these technologies were developed and people realized they could apply them to sequence bacterial populations, literally the whole field changed. Because suddenly it became feasible to say, I'm going to look at the entire human body and identify what bacteria are present. Or I'm going to look at people that have Crohn's disease and people that don't and see what differences there are. Or I'm going to sequence the microbial population of 15 places across Yellowstone and see which microbes are growing in which spaces, and do that in a number of weeks really inexpensively. So this, I cannot overestimate how important this is to everything, this technology is to everything else that we're going to talk about. It undergirds everything. Now, that's about the extent of genetics that we're going to talk about for the next little while. I want you to understand this underpins everything. We use the genetic analysis as the identification tool. So everything that you hear me talk about going forward about how we've identified these bacteria being present is using this genetic approach to do that. Everybody tracking with me? All right. So the Human Microbiome Project was a 5-year federally funded project, much like the Human Genome Project. And scientists said, we're going to gather a whole group. I think there were 80 different scientists working on this, each working on different parts of the body. And they took 250 healthy individuals and they sampled the microbiome on and in their body. So healthy is really important, because they had to show that they didn't have disease, and that includes gum disease, because about half of them had gum disease and had to be treated for gum disease so that they knew that the bacteria present in their mouth were not disease-causing bacteria. So healthy individuals, and they sampled them at somewhere between 15 and 18 sites. There were additional sites sampled in women because of the length of the reproductive tract than in men, but there were lots of samples taken from the mouth, the cheeks, the tongue, the scrapings of the teeth. We're going to talk about that in a couple of weeks. The skin, multiple places, behind your ear, on this little fold on the side of your nose, in the corner of your elbow, on your back. At the beginning of the crease, right, well, you know what I'm talking about. Lots and lots of places that they sampled the human body. Multiple places in the reproductive tract, and then also again in the gastrointestinal, the digestive tract. Many of these people were resampled at multiple times. So if they got sick and were on an antibiotic, they were resampled. If the women got pregnant, they were resampled. If they gave birth, they were resampled. So you can begin to track and chart if the microbiome is very, very stable or if the bacterial populations ebb and flow. And they use the DNA sequencing process that we talked about on the previous slide. Their initial findings were published in June of 2012. There were somewhere in the neighborhood of 20 or 30 papers published simultaneously across multiple journals. It was a really exciting collaboration between journals that didn't necessarily talk to each other historically, that all were agreeing to publish information at the same time, different aspects of the information. They identified over 10,000 different species of bacteria that live in and on the human body, and each one of us has about 1,000 of that 10,000. So right away, you know, there's a fair amount of variation, a lot of differences, and we'll talk more about that. 100 trillion, now we're talking primarily bacteria, 100 trillion bacterial cells in the human body. Now you may have heard that there are 10 times as many bacterial cells as there are human cells. And until about 2 months ago, that would have been completely true. But we realized about 2 months ago that we were undercounting the number of human cells. And that humans didn't have 10 trillion cells. Humans had about 37 trillion cells. I don't know where the other 27 trillion were hiding. That is really difficult for me to get my brain around. But at the end of the day, what it means is that bacteria outnumber human cells about 3 to 1. That's about 2.5 pounds of bacteria living in and on your body. That's almost the weight of your brain. And it's about 3 pints of bacteria, if you want to think about volume, because these 100 trillion bacteria are tiny. That amazes me. 100 trillion bacteria in 3-pint glasses. That's pretty impressive. So you can congratulate yourself because you are a superorganism. You are truly more than the sum of just your cells. There are an enormous number of other organisms making your home a fantastic place for them to live and also giving you benefits as well. Again, resist the urge to pull out the hand sanitizer. Resist the urge to run home screaming with Lysol and spray everywhere. They are beneficial by and large. We're going to probably see lots of images like this, so let me take a little bit of time to walk you through what's on this graph. For the Human Microbiome Project, they sampled 250 individuals at multiple places around the body. And then they figured out, okay, what bacterial populations are living in the mouth of sample 1, and in the mouth of sample 2, and in the mouth of sample 100. And how do we actually compare the mouth samples between all these patients to the samples in the arm or the samples in the nose? How do we do that? So they use something called principal components. I don't understand principal components very well, but in a nutshell, here's the concept. It's a mathematical way of comparing two populations. And you essentially choose the parameters of what you want to compare, and they form your x and your y axis. And so when you're looking at two populations, the closer they are on a graph, so the closer their x and y coordinates, the more similarity is present in the population at each of those samples. So in other words, on this image on the screen, if you see two dots that are very, very close to each other, it means that the bacterial populations at each of those samples are very, very similar. If you see two dots that are very far apart, it means that there are very different bacterial populations in those two samples. So what is amazing to me is that when you look at the 250 individuals and you look at the samples from the mouth and the samples from the skin and the samples from the nose and from the gut, you find that there is a lot of similarity when I look at organ system or location of the microbiome across my population. In other words, the people that I sampled, all of the bacterial populations from their mouths are very similar to each other. That's this upper bluish at the upper left-hand corner of the screen. So these are all, oh, I'm sorry, that's the gastrointestinal. Let's just go with that. Here's the gastrointestinal. We're going to go with the gastrointestinal. So all the gastrointestinal samples, y'all knew that, didn't you, are very, very similar from patient 1 to sample 65. They have similar sets of bacteria. But look at how far apart they are on this graph from the bacteria that are present in the mouth, the oral. And all the orals roughly cluster together. And they cluster differently from the skin. And there's an overlap between the skin and the nose, which perhaps is not necessarily so surprising, but they're distinct from the urogenital in many ways. So if you look and try to say, well, do they cluster by males and females? Do they cluster by young and old? Do they cluster by socioeconomic group? No, they cluster by body type. So if I were to look at the microbiome from your skin, it is much more similar to your neighbor's skin microbiome than your skin microbiome is to your gut microbiome. So there are these distinct communities of bacteria that we find predominantly in the gut, or predominantly in the skin, or predominantly in other places. Let me stop a second and see if we've got questions.
What are the percentage values on the axes?
Yeah, so Grace, that's a really good question. I can't tell you what the percentage values on the axis mean. I'm not 100% sure. I know it has something to do with the tolerance that you're allowing, but don't write that down because I might be really, really wrong. So, okay, so now someone's asked the first question and I couldn't answer it, so everybody can ask questions now. Pressure's off. Any others? All right. Is there consistency across cultural groups? The consistency across cultural groups, I'm going to say this and I'm going to give you a caveat. The consistency across cultural groups doesn't seem to be any different than the consistency between cultural groups. So if I take an individual from Africa and look at their skin microbiome, it's going to be very similar to an individual from Asia's skin microbiome. Now the caveat that I'm going to give you is when we get to the gut microbiome, because that's driven very heavily by diet. And diet is different in multiple different cultures. But by and large, you have a very similar microbiome to a person that's living on the other side of the planet. Let me see if there's any questions that are coming in from my online folks. No questions yet. Any others? How different the oral microbiome is from the gastrointestinal, because they're at two ends of the same tube. Yeah. There are very different bacteria that carry out a certain function in the mouth than in the gut. So the conditions are very different. It's a much higher oxygen content in the mouth. Many parts of the gut have almost no oxygen. The pH is a little bit different. The types of food molecules are much more complex, haven't been broken down in the mouth than in the gut. So there are pretty significant differences, even though they're both parts of the digestive tract. Mark?
Do the outliers have stories behind them? For example, we've got one little skin one that's right in the middle of the oral.
Yeah, and there's one right there. Yeah, do the outliers? That's a good question. The paper doesn't dig into the individual outliers. There are some anecdotal stories about some of the outliers that seem to suggest that there are some uniqueness, maybe some really unusual diets or some interesting family histories. And this is another place where correlation and causation are going to be tricky, because many of the stories that I'm going to tell you are stories of one. And while they're really intriguing and we can draw some interesting conclusions, we can't generalize. But yeah, I would love to know some details about this individual up here whose nasal microbiome is very similar to the gastrointestinal tract. Yeah, we're not going to go, I'm not going to take us down that road. Although I will say it does bring interesting meaning to the phrase brown noser, but we're just going to stop that right there. I'm sorry. I told you I was not going there, and I sure enough did. Let me take one more question, Bob, and then we'll move on.
That's a two-dimensional scatter chart.
Yes, it is a two-dimensional scatter chart.
Are there only two principal components in bacteria?
Are there only two principal components in bacteria? So we are now past my knowledge on principal component analysis. I would be really happy for somebody who knows principal component analysis and bacteria to give me some background on that this week. That'll be something that I will definitely be working my way through. Okay, so let's move on. All right, let's talk just for a second about the skin. I'm not going to spend a lot of time on this. This is a busy slide. I recognize that. I don't want you to dig into details. I want you to appreciate the overall piece. Each one of these circles on the right-hand side of the screen represents the specific populations of bacteria that are present at that specific skin site across all the individuals that were sampled for this study. Now, let me back up a second. Do you remember kingdom, phyla, class, class, order, family, for green salad, genus, species? Back to your biology taxonomy. King Philip called out for green salad was always the way that I worked to remember that. It's a way that we classify organisms. You and I probably went to high school in a time when we had a three-kingdom system that was plants, animals, and then the Monera, which was this bacteria, archaebacteria kind of situation. That's all changed. We know a whole lot more from classification now. We're going to focus just on the bacteria, the kingdom of bacteria. The phyla is a smaller subgroup of classifications, and then the order, the family is below that. Most of what I'm going to talk to you about is either going to be the phyla, or I'm going to talk some about the family. It's just a way to broadly categorize. So you're not going to hear me, for example, talk about Escherichia coli very often. Instead, you're going to hear me talk about the higher level, which I think is the actinobacteria, which is the phyla that Escherichia coli lives under. There are lots of bacteria that live under these broad categories. So when you see 6 or 7 colors and you see actinobacteria, bacteroides, firmicutes, these are broad headings with thousands of bacteria underneath them. But the point about this slide is that each one of these sites has a different population of bacteria. And it's all from your skin, but some of your skin is like a desert. It is incredibly dry. Skin on your arms and legs is more exposed to the environment, and so you have a much different diversity of bacteria on those places than you have, for example, under your arm, or in places where it's moist, or places where it's waxy like your forehead. So there are different populations of bacteria. Now here's what I find really interesting, this next image that's on the left-hand slide. These are 4 different individuals that are sampled. So the antecubital fossa is the crease of your elbow. The back, the nair, which is your nose, and the plantar heel, the bottom of your foot. So these 4 individuals were sampled, individuals 1, 2, 3, and 4 are H1, H2, H3, and H4, at these 4 locations. And what you can see right off is that there's a lot of similarity between those 4 individuals within each part of the skin, but there are big differences between the skin in the crease of the elbow and the skin in the nose and the skin in the back and the skin on the bottom of the foot. So very, very different populations. You can think of this like ecosystems. There are animals and plants that thrive in moist rainforests. It's a totally different set of animals and plants in the rainforest than in the desert tundra or in the Arctic. Same sort of thing on your skin. Lots and lots of different ecosystems on the skin. All right, so back to the Human Genome Project. Incredible amounts of diversity in the human genome. It's estimated that if you looked at all the genes present in the bacteria of the human, living on the human, 8 million genes compared to the 20,000 or so that we have in our DNA, in our cells. A huge population of genes. This is valuable because it means we have access to all these genes. Well, what does that mean? Well, let me tell you. You and I have 20 genes in our human cells that encode enzymes. So genes encode recipes for proteins. An enzyme is a type of protein that carries out a function. Enzymes that break down carbohydrates. These are things like starch, but they're also things like the type of molecule that's found in celery. We don't have a whole lot of genes for digesting celery. It's roughage. It just passes right through our body. This one type of bacteroides, and I don't even know how to pronounce Theta has 260 genes that digest different kinds of carbohydrates. I can bet you it has a gene that produces an enzyme that can break down celery. Why is this important? Because if you have these bacteria living in your gut and you eat celery, these bacteria can break down celery, break down those carbohydrates, and turn it into energy that your body then can use. And if you are looking for whatever food source you have and you don't know where your next meal is coming from, it's a good thing that you have bacteria living in your gut that can break down celery. Now, if you are around food all the time and food is plentiful and you have bacteria that are extracting extra calories out of celery, that may not be such a good thing. And that's part of the challenge of the microbiome that we've had for tens of thousands of years, butting up against an environment that has very rapidly changed. And we'll dig into that in week 3. There's a fair amount of variation between individuals that we talked about, but there's this concept of key species, which functionally means that Mark may have a specific bacteria in his gut that has an enzyme that digests these carbohydrates. I may not have that same bacteria, but I may have a different bacteria that also digests carbohydrates. So functionally, we've got that box checked, even though they are slightly different bacteria. So functionally, we cover most of the key pieces, even though it may be individually different bacteria in that slot for each one of us. And this whole concept is what we're going to heavily talk about, the personalized microbiome. How is my microbiome from my skin, from my gut, from my mouth, how is it different from yours? And it's shaped by early life experiences, the way we're born, our diet, exposures that we've had throughout life. And that's how it becomes this very personal microbiome. All right, so we've talked through a little bit of the background. Let's now dive into some stories. There's a fantastic article in the New York Times in June, written by Carl Zimmer. It's called Tending the Body's Microbial Garden. I would encourage you to go take a look at the New York Times Tending the Body's Microbial Garden, because there's this great image, which is, I recognize, a little hard to see here. So let me just tell you broad pictures. This is a visual of the human microbiome. The picture in the center, this is the family tree of bacteria. It's quite a convoluted, complicated family tree with lots of branches and lots of individual types of bacteria, shown in this nice circular window. And then the concentric circles that come out from around it are the bacteria that are those specific bacteria present in the different parts of the body. So here are the bacteria that are present in the mouth, on the cheeks, on the teeth, on the tongue, and you can see they're spread out across the entire family tree of bacteria. So there are lots of different types of bacteria that are present in the mouth. And by and large, if you find it on the teeth, you're going to find it on the tongue, and you're going to find it on the cheeks. That's not 100% certain, but there's a lot of similarity. Here's your gut microbiome. This is tricky to see because it is really faded, but on the right-hand side, there are blue boxes. You can probably see this better on your handout. And on the left-hand side, down at about maybe 7 to 9 o'clock, is another set of blue boxes. These are the two broad classes of bacteria that inhabit your gut, the Firmicutes and the Bacteroidetes. You will not be required to know how to pronounce these, but you will hear me talk a lot about the Firmicutes and the Bacteroidetes, because these are the two primary populations that live in your gut in different frequencies. And some data suggest that when this balance of Bacteroidetes to Firmicutes is out of whack, then there are problems with the gut and problems with the metabolism and problems with weight. So we'll dig into that a little bit more. But two big groups, Bacteroidetes and Firmicutes. How do you get your microbiome? The party line has been for a long time that the fetus is sterile, that there are no bacteria in the embryonic or in the fetal environment. That is starting to change because we're starting to see some evidence that there may in fact be bacteria that cross through the placenta or cross the umbilical cord that may early populate the fetus. The jury's still out on that, so hang with me on that. Okay, I'm moving into inappropriate territory. Your first dose of microbes, your microbiome, comes from how you were born. And for the vast majority of people, it comes on your journey through the birth canal and through the female reproductive tract. Now, those of you in the audience may be asking, what about cesarean sections? Hold with me, we'll talk about that in just a second, I promise. But the first dose of bacteria that an infant gets is ingested as they travel down the reproductive tract. This piece that I'm about to tell you, I find amazing. During pregnancy, the populations of bacteria that are present in the vaginal tract of a woman change, starting at about the second trimester. There's a population of bacteria, lactobacillus, that become much, much more prevalent, that are almost never seen in the vaginal tract until this period in pregnancy. And after birth, they disappear. So this is a primary population of bacteria that an infant takes in. Lactobacillus produce an enzyme that helps them break down lactose, which is a sugar found in formula and breast milk. You and I have a gene in our human cells that makes an enzyme that helps break this milk sugar down as well. But if the infant starts out with a big population of these bacteria, there's an extra boost and there's an extra amount of energy that they can get from their feedings. I find this fascinating, that the body knows how to prepare the infant for the food tasks that are at hand. So there are lots of things that seem to influence how you got your first microbiome. Where did you get your microbiome? Here are many of them. The data is still out on some of them, so some of these are a little shakier than others. I'm going to focus on the mode of delivery, the mode of feeding, and what happens when you're given other foods and fluids. We'll talk about antibiotics the last week. So these are the ones that we're going to talk about. I do find it interesting, the maternity ward or neonatal unit, which has to do with how many other kids are around you and how many other nurses handle you. Where does your microbiome come from? Again, I want you to be careful. I don't want you to envision putting on gloves to create a sterile environment. We need a microbiome. Infants need a good, solid microbiome. If you don't have a microbiome, we see this in mice, you are not necessarily healthy. If you have a little bit of a microbiome but not a full, diverse microbiome, there are challenges and problems, and we'll dive into that too. All right, let's talk about mode of delivery. So, I've already told you that if you go through a vaginal birth, you are likely to pick up the microbiome from the reproductive tract. You're also likely to pick up a little bit of the microbiome from the gastrointestinal tract, because there's a little bit of mixing during the birthing process. This is a really important source of lactobacillus, which we just talked about, and a couple of other key bacterial groups. So an infant's early gut microbiome looks almost identical to the reproductive tract microbiome of its mother. And then over time, it shifts and changes to look more like the adult gastrointestinal tract. Infants that are born like me by cesarean section, their bacteria that colonize their intestine are not necessarily maternally derived. They don't pick up that maternal reproductive tract microbiome. They pick up a microbiome that looks a whole lot like skin, which is not surprising because they're being handled, they're putting things in their mouth. If they're nursing, they're picking up the microbiome from the skin of the breast. So their early microbiome looks like skin, which is a very different microbiome than the infant microbiome that looks more like the reproductive tract. So the colonization of some of these key bacteria, like lactobacillus, gets delayed. Now, I didn't say never happens. It gets delayed. But it may take 6 to 7 months before the microbiome of an infant born by cesarean section looks like the microbiome of an infant born vaginally. That's a pretty significant difference. There are a number of parents that are now learning about the microbiome that, if they know they're going to have a cesarean section, will actually have swabs at the ready so that they can inoculate their infant at birth with secretions, with microbiomes from the reproductive tract. Now, let me back up. There's a lot of studies out there that are saying, what about women that start labor and partway through labor actually have to have a cesarean section versus women that never start labor and have a cesarean section? There are some differences in the microbiome there as well because the infant has, at least in some cases, engaged partly into the birth canal. So there are some nuances about this as well. Are there long-term impacts? There have been enormous numbers of studies looking at cesarean births and vaginal births, and there's a higher incidence of things like eczema, hay fever, asthma, type 1 diabetes, food and other, sorry, there was a word missing, food and other allergies from cesarean births. Again, correlation does not necessarily equal causation because many of these women are given antibiotics before delivery. And we know antibiotics change the microbiome. So these infants may have a different microbiome and may be more susceptible to some of these disorders because of the antibiotics, not necessarily because they were born by cesarean section. So stay tuned on this. And again, this is another one of those no guilt. You don't have a choice in some of these issues. You know, I'm thankful that I just happened to be here, which I might not have been if I was not a cesarean section kid. So, you know, there are trade-offs. Okay, let's take a break. You have 10 minutes for snack for those fantastic cookies and coffee. Those of you that are watching this online, hang with us and come back in about 10 minutes. Let's get started again. I have 2 questions from our online audience. Let me go ahead and answer those. The first one is, with the advances in sequencing, do you see more advancement in treating infections and disease? So with new technologies that allow us to sequence more quickly and more rapidly, will that have any impact on treating infection and disease? I think that it actually will. In fact, it already does because it allows us to track infections much, much more quickly. So when we have outbreaks of infections, we now can very rapidly track the specific mutations, the specific strain causing the infections, and watch as it, well, not watch, but follow it as it moves from person to person and as it gains new mutations. There's a couple companies at HudsonAlpha that are actively working in this. Jen Hahn, one of our faculty researchers, does a lot of work in this area. So the new technology lets us track, and as we see how the infections mutate, it gives us a sense of how they might evade or might become resistant to drugs, and we can identify bacteria that are on the verge of becoming highly resistant in advance of the resistance actually developing. Great question. And then I have another question that says, I am all for cleanliness, but is society's obsession with killing bacteria causing more harm in the long run? The short answer is quite likely yes. We'll dig into this a little bit more either tonight or potentially first thing next week, depending on how far we get. But the idea that the only good bacteria is a dead bacteria has some real problems with it. And when we wipe out bacteria from our system, we're oftentimes having unintended consequences because either those bacteria had an important benefit or we're now making room for not so pleasant bacteria to move into their place. Good questions from our online group. So we talked about method of birth. Now we're going to talk about method of feeding. And I know this is super controversial. I recognize that. But let me just talk through this. Breast milk versus formula. There are several hundred species of bacteria present in breast milk. Several hundred. This is not bad. These are valuable bacteria. These are different from the bacteria that are present on the skin. So it's not contamination from the skin, from the breast skin itself. The thinking is that many of these bacteria actually help seed the new gut microbiome, the gut microbiome of the infant. In addition, the colostrum, which is the clear liquid that is first to form right after the infant's born, that's delivered from the breast, the colostrum has more bacterial diversity than mature milk. So the biggest bolus of different bacteria happens in that early delivery of the colostrum, but there still is bacteria present in mature milk. Milk also contains prebiotics. Now, we haven't talked about prebiotics, and let me just introduce it to you, but we're going to cover it in more detail in week 4. A prebiotic is some substance, usually potentially a food source, a nutritional source that helps bacteria to grow. That's different from a probiotic, which is an actual bacteria that's thought to be beneficial. So prebiotics set the stage to allow the growth of bacteria. Probiotics are actual good, and I've used that loosely, good bacteria. So breast milk has a lot of what we think are prebiotics that encourage the growth of other bacteria. Now, so what if we compare the microbiome of infants fed with breast milk versus the microbiome of infants fed with formula? So many of these studies were first done several years ago before we had, number one, the new technologies, so they were based on what you could culture, what you could grow on an agar plate. And number two, infant formula has changed pretty radically over the last 30 years. So the early studies found a lot of difference in the bacteria that were present in infants that were breastfed versus infants that were bottle-fed. And the more recent studies do find differences. So they find that there are more of a type of bacteria called bifidobacteria in the microbiome of infants that are breastfed. This is the gut microbiome of these infants. And there are more Bacteroidetes and Lactobacillus found in the infants that are formula-fed. So clearly there is some difference in the bacterial makeup. But these differences are relatively slight, which suggests a couple things. One of them is that the more recent studies that show the slight differences potentially reflect the fact that infant formula now more closely mimics the nutritional components of breast milk. So by mimicking the nutritional components of breast milk, it is allowing the very similar populations of bacteria to thrive. Now, infant formula still doesn't carry the antibodies that you're going to find in the breast milk, but that's a different scenario. We're just talking about the bacteria that are growing in the gut. So there are some differences in the microbiome of infants that are breastfed versus bottle-fed, but it is not nearly as dramatic as the differences between infants that are vaginally born versus born by cesarean section. Now, this is the major driver of what shifts an infant's microbiome. This is the driver of your microbiomes as well. And that's when you introduce solid foods. The moment you introduce rice, peas, fruits, grains, you now set up a whole new environment. And some of the microbiome, some of the bacteria that thrived under formula or breast milk aren't getting the nutrients they need to survive. And other bacteria that were just barely hanging on now have a feast and are able to really grow and develop. So I'm going to show you a study that was done comparing the microbes of the gut from children in Burkina Faso and Europe. Now, I didn't know where Burkina Faso was. Many of you may know, but here's a map to help you. Burkina Faso is in Africa. It is around Mali and Niger and near Ghana. And the European kids in this study primarily came from Italy. So it's important to know that these infants, these children were followed when they were, and they were almost, regardless of what population they came from, they were both primarily breastfed. And the microbiome of these kids from Burkina Faso and from Italy were pretty similar when they were being breastfed. And then when they're weaned, they're then put on the diet, the traditional diet of their populations, which is dramatically different. If you live in Burkina Faso, you have a diet that has almost no fat and animal protein, is very, very heavy in vegetables and cereal grains. It is extensively pretty much vegetarian except for the occasional termite, which is an important protein source for these individuals. This is not the paleo diet. So there are a lot of nutritional deficiencies in these kids as they grow, but that's their diet compared to the traditional Western diet of kids in Italy, which is high in fat, high in animal protein, high in sugar, high in starch, and low in fiber. This is the typical macaroni and cheese. Well, probably not in Italy, but you know, So at the moment that they started being fed this kind of food, they started noticing major differences in their diets. Now, remember I told you that the gut microbiome has two primary species, two primary phylum, two primary groups of bacteria, the Bacteroidetes and the Firmicutes. You're going to see in the next slide the ratio of Bacteroidetes to Firmicutes in the kids from Burkina Faso and the kids from Italy. And they are dramatically different. So the green parts of the circle are the Bacteroidetes and the red are the Firmicutes. And you can see that the kids on the right from Burkina Faso, most of their gut microbiome are these Bacteroidetes bacteria types, which is almost the exact opposite of the gut microbiome of the kids from Italy, where more than half of their microbiome are these Firmicutes. So clearly one of them is doing well on plants and the Burkina Faso, these Bacteroidetes do really well on plant and vegetable matter. And the Firmicutes are pretty heavily dependent upon high-fat, high-protein diet, which you got a lot of in your European group. So your early diet is a huge driver of how your microbiome ultimately develops. I love this image. This must be a mom who was in science studying the microbiome and was pregnant and realized that she had a phenomenal built-in microbiome analysis with her son. This is an infant who was followed for the first 2.5 years. So essentially, she brought his diapers in quite periodically and said, let's look at the gut microbiome today. So each one of these bars running from left to right is a sample of the gut microbiome from a different day, all the way up to like 834 days. So clearly I'm not showing you all 834, I'm just showing you representative samples. The specific colors that make up each bar are the classes of bacteria that are found in the infant's gut on those specific sampling days. And you can see we start out with mostly this pink color, which are the Firmicutes, which include our lactobacillus. So not surprising based on what I've just told you. Around day 92, the infant has a fever, and you see a rise in these light blue and these dark blue, the proteobacteria and the actinobacteria, probably which has something to do with the fever and with the infection around that. Then around day 168, we introduce table foods, and you now see the growth of these yellow boxes, which are the Bacteroidetes, because we're feeding the kid peas and carrots and vegetables. And we just know, I just showed you from the previous slide from the Burkina Faso, the Bacteroidetes thrive on vegetable material. And then over here around day 377, there's another illness and the child's put on an antibiotic. And look at the difference in the microbiome from the box just before day 377, just outside of the red box, and the first bar, the bar right inside and the bar right next to it. We lose an enormous amount of diversity, in part because of the antibiotic well, let me back up. Potentially, causation is not correlation. Potentially because of this antibiotic. So there's a loss of bacterial diversity that takes several weeks before it ultimately recovers. We're going to have to see many, many more longitudinal studies like this before we can specifically say this bacteria changes when you have this population. But this kind of study is fascinating to watch how the day-to-day interactions, the food we eat, the illnesses, the medicines that we take, how they change our microbiome of our gut. Yeah, wow, cool. Who ever thought that talking about poop would be so interesting? Okay, I love this story. This is a story from a group out of Sweden, and it's a small study, and I'm going to tell you about some challenges. This falls in that category of don't do this at home yet. So this was a study of allergies using about 184 babies from Sweden. And the researchers asked lots and lots of questions to the kids asked lots of questions about the kids to the parents during multiple years. It was a multi-year study. And they followed which kids developed allergies and eczema. Eczema is an early indicator that a child is likely to develop allergies. That is certainly true of one of my children. We saw eczema, and she has all kinds of pollen and hay fever allergies. So they looked to see, do kids have eczema at 18 months and 36 months? And can we track that back to anything that's happening earlier in life? One of the questions they asked was, what do you do when the child's pacifier falls out of his or her mouth? And a large portion said, I rinse it off with water and pop it back in. But about a third of parents say, I put it in my mouth, rinse it off, and put it back in my child's mouth. Okay. But here's the punchline. Those kids whose parents were a lot less likely to have eczema, a lot less likely to have eczema. So the question, or the theory, hypothesis, is that maybe what happens is these parents are transferring some of the bacteria, the healthy bacteria from their mouth, and as the child, and I'm going to give you some details in just a second about how our growing early microbiome influences our immune system. So maybe taking some of these healthy bacteria from mom or from dad is actually helping protect them against allergies and autoimmune diseases. So this may help train the immune system. So a big study came out about this. I'm sorry, let me back up. When this study was published, all kinds of people wrote in. Dental hygienists wrote in, and pediatricians wrote in and said, there are all kinds of bacteria in an adult's mouth that are harmful, that cause gingivitis and tooth decay, and you don't want to put that in an infant's mouth. This is a really bad idea. Point taken. That's part of why I'm telling you, don't do this at home. There was a report about it on NPR. And this woman, Amanda Sauer from Washington, D.C., uses a pacifier for her 2-year-old son when he gets fussy. And when the pacifier falls out, she rinses it off. And she said that she's not likely to actually put this in her own mouth, partly because she doesn't want to put a pacifier in her mouth. But here is my favorite part of this story that leads us into where we're ultimately going. But sometimes the dog cleans it off for us. So maybe that's just as good. Yeah. So we're not getting the human microbiome, but we're getting the dog oral microbiome. Yeah. I'm willing to bet that she is not the only parent that is experiencing this. But this is a perfect lead-in for where I want us to go next. And this is going to lay the foundation for why we think what happens in our early immune system, in our microbiome, our early microbiome, plays a huge role in the development of our immune system. We will probably not get through all of this tonight, but I want us to get as far into it as we can. So let me take a very quick second and see. All right, no questions from our online audience. A couple questions from you guys. Anybody? Shirley. Oh, what a good question. Could you use any breast milk? I don't know. I have not seen those studies. There's a really interesting study there because there are breast milk banks where women who produce extra milk are actually giving or selling it to women that want their children to have breast milk but for whatever reason can't produce their own. And there are lots of questions about how is that being screened? Is that being checked to make sure there are no harmful bacteria in that? That is an excellent question. Is there some special benefit from the mom who has genetic information being shared with her child versus another? I don't know. That's a really cool question. I'll try to see if I can find that out in the next week and report back. One more. Ooh, I got like 16 hands up. Sir, you were the first one up. You were right there. You were Johnny on the spot. Ooh, pasteurization of milk, and pasteurization to kill bacteria that are present in the milk naturally to give it a longer shelf life. Yeah, you could make the argument, as I'm going to walk through some of these pieces in a minute, that pasteurizing milk potentially reduces our microbial diversity. Now, some of that is an issue because most of us don't have cows in our backyard. So if we want milk, we have to get milk that has some sort of shelf life. But that's a really interesting point. What are we now drinking what were we drinking with unpasteurized milk that we are no longer gaining when we drink pasteurized milk? Really good question. I love this audience. You guys are fantastic. All right, let's move on. So let's talk about microbes and your immune system, because this is going to play an important role in some of our discussions over the next several weeks. Here's the premise. Exposure to germs, and I should not use that word germ, I apologize, because I really should use the word microbe, because germ has a negative connotation. Exposure to microbes as a child seems to be beneficial, whereas living in a microbe-free, and in reality none of us live in a microbe-free environment. There are microbes everywhere. You cannot kill all the microbes. But if you wipe out lots of the microbes and you live in a less diverse population, that seems to carry some health risks along with it. So this really stems from a finding in 1989 looking at incidences of hay fever and eczema, and finding that they were less common among children that came from large families, that had lots of siblings. And a study that was released shortly thereafter that found that kids that grew up on farms or around lots of animals also were less likely to have eczema and hay fever. And then another study looking specifically at the nation of Ghana, as Ghana went from a developing nation to a more developed nation, and as hygiene improved and water quality improved and houses had floors that you could clean rather than dirt floors, found that as the nation became more industrialized and presumably more hygienic, immunologic and autoimmune disorders, things like lupus, type 1 diabetes, increased as the nation became more developed. Those pieces all together form something that was called the hygiene hypothesis. Now, I'm going to tell you what the hygiene hypothesis is, and then I'm going to tell you what the modifications have been made to it in the last 4 or 5 years. The hygiene hypothesis proposed, based in part on graphs like this, that showed the incidence on the left of infectious diseases, rheumatic fever, hepatitis, tuberculosis, mumps and measles, dramatically dropping from the 50s to 2000, and at the same time, things like multiple sclerosis, Crohn's disease, type 1 diabetes, and asthma dramatically increasing. Now, what rule are we going to apply to this right off the bat? Correlation is not causation. But there is something to explore here. And the hygiene hypothesis suggests that as kids were less exposed to childhood infections, they had an increase in things where their immune system was turning on itself. Allergies, food allergies, as well as hay fever and being allergic to pet dander, and things like autoimmune diseases. So the theory was something like industrialized patterns of living and vaccinations did a great job at reducing the infections, the left side of this image, but led to an unintended rise in allergy and autoimmune disorders because the body now had fewer and fewer things to early on prime the immune system to begin to recognize, and as a result, the immune system wasn't regulated properly and attacked itself or attacked things that it saw coming through the digestive system like peanuts or eggs, and led to an increase in allergies. Now, unfortunately, this whole concept of hygiene got transmitted to the public as being too clean, that we were too clean. And that kind of is a distractor. That's not the point of this was kids were getting less infections, and so was that causing their immune system to be poorly regulated? Some additional work on this has suggested it is not being exposed to these childhood illnesses. So it's taken the infection piece off the table. Whether you're exposed to measles, mumps, rubella, rheumatic fever, whether you get the vaccines or not, doesn't seem to be the issue here. The issue seems to be something slightly different. So the theory has been modified to be called the old friends hypothesis. And we're going to walk through what the old friends hypothesis means. So it refocuses the causation away from these infectious microbes and looks instead at those bacteria that have been part of our lifestyle and have taken up residence in our body for tens of thousands of years. These have been historically well tolerated by our system. We don't have to go back too many generations to find our ancestors that lived in dirt. That dirt was everywhere. Dirt was on our food. Dirt was, we played in the dirt. You put dirt in your mouth. You were around lots of animals. You had big families. There were lots of microbes all around you. That's very different than the world that we live in today, where we have less and less chance to interact with microbes. And I'm not talking about harmful microbes. I'm talking about just everyday normal bacteria that take up residence with us. So let me show you this image, this illustration. I want you to focus on the top half with the prototypical caveman. Think about life in that window, and you don't have to go back to caveman times. You can drop back, 100 years or less. No refrigeration. So there were many more bacteria living in food, and that's not necessarily all harmful bacteria, but refrigeration slows the growth of bacteria. No antimicrobial. There were no antibacterial anythings. None. There was no hand sanitizer. There was no soap. There were no any of these things. The water wasn't chlorinated, so there were a whole lot of things living in the water. Sometimes not so good for you, but a lot of times they just existed in your gut. Almost exclusive breastfeeding. The foods were mainly raw, or they were pickled or fermented without a lot of antibiotics. And almost everybody was born, just about everybody was born vaginally. Today, we have refrigeration, widespread antimicrobial, chlorinated water, a lot of folks that formula feed, refined foods. Very rarely do we eat fermented or pickled foods, and lots of cesarean births. Now, I am not saying this is bad. Hear me. I would not be here, which therefore means you would not be here tonight if we did not have cesarean section births. I like my food to be refrigerated, to not have as many microbes, and I like chlorine in my water. But the reality is, as we've shifted, there's been less and less microbes that we find ourselves continually exposed to. So our overall microbial diversity drops. So how does this actually fit in with our gut? Why are we talking about this? Okay, your digestive system is both, your gut is both a digestive organ and an immune organ. Your digestive tract is full of these little folds, these little villi, little finger-like projections that line your digestive system. In fact, if you were to spread out all of the surface area of your digestive system, one person's digestive system, it would cover a tennis court. Yeah, that is a lot of surface area. And your gut has more microbes per square inch than almost any other system studied around the world. Lots and lots of microbes, lots and lots of gut space. Now, one specific part of your gut that I'm going to focus a little bit on are these pieces called the Peyer's patch. They look almost like little domes that are just below the level of the villi. So this next image shows you a cross-section of Peyer's patch. Your gut, Peyer's patches, and your villi not only absorb nutrients, but they are a gathering place for your immune system cells, your B cells, your T cells, all those pieces to hang out. And like this immune cell is doing, and let me show you in this enlarged image, this immune cell is sending up a projection that is actually sampling what is present in the fluid floating past the digestive system. So this is your food, this is some of your bacteria, and so it periodically up periscope grabs a sample and comes back down and says, what is this? What's hanging out in my digestive system? And based on what it finds, it either says, oh, okay, I know this. This is me. These are just cells being sloughed off from me. Or, oh, this is friendly. These are those bacteria that hang out. I've seen these really for, since I was young. Oh, there's a peanut piece. Okay, oh, there's some egg. These are okay. Or it says, ooh, I don't recognize this. This is foreign. This is bad. I need to mount an immune response. So the microbes that are present in your gut at some standard level, the diversity of your microbes, help your immune system train to know, I see this a lot, this is okay, or I've not seen this before, this is problematic. The theory, yeah, I love this part, heirloom strains like heirloom tomatoes. So the old friends theory is that there are heirloom bacteria that have been around in our gut for tens of thousands of years, but because of the change in the way we live, as we reduce the diversity of our microbes, we reduce the ability of our body. Our body has fewer and fewer of these heirloom strains and is less and less likely to appropriately regulate itself. So, because there are fewer things in the gut, it doesn't train itself properly and says, oh, that's a peanut, that's bad, I need to mount an immune response against that. Oh, that's one of my own cells, that's bad, I need to mount an immune response against that. Let's go travel and kill those cells. That's the concept of the old friends theory. Now, I'm not going to dig into this piece because this is a little bit more advanced than I want us to go into right now, but we actually now have evidence that there are specific molecules in our gut that look at these things that are floating by our digestive system and say, do we attack or do we stand down? And that there are differences in how those are responding in individuals that have less diverse microbiomes than individuals that have a very rich diversity of microbiome. Before you run out and start eating dirt, before you run out and wipe and buy every probiotic in the grocery store, hang with me till we get to the end. There's a lot of evidence behind this theory, but this is still very much correlation equals, is not necessarily causation. So, here's next week's topic. Last slide. We're going to start by talking about the microbiome in your home. Again, do not panic. Do not panic. And then we're going to start talking about diseases that may be a consequence of this disturbance in our microbiomes, especially in our gut. We'll talk a little bit about malnourishment, and then we're going to move into behavior, some brand new work that's really intriguing in autism, some cardiovascular and cancer links to the microbiome, and then we're going to end up with how your microbiome, I told you how it forms, I told you infant to early toddlerhood, now let's talk about how stable is it over adulthood, and what does it begin to look like as we age. So, that ends week 1. I know that we have just covered a ton of information, a lot of information. Take them home, look at the notes, read through the FAQ. I hope to see you back next week. Drive home safely tonight, everybody. Take care.